Today we're going to be going over chapter 10, which we're going to be talking about the structure and function of DNA. So in the past we were talking about inheritance and how we know things get passed on from one generation to the next. Now we're going to actually look at how DNA is structured and how it functions as well.
All right, so for us to understand the structure and purpose of our DNA, we've got to learn more about the specific bases. We talked a little bit about those back in chapter 3 when we looked at nucleotides, my goodness, and nucleic acids and how they make up DNA and RNA. And those nucleotides, we actually have two different types of nucleotides. They're either purines or pyrimidines. And this is going to be based off of if their nitrogenous base has a two-ring structure or a single-ring structure. And within DNA, there's 4, and with RNA, there's 4 as well. So we talked about those 4. So in our purines for DNA, we have adenine and guanine. And our pyrimidines for DNA, you have thymine and you have cytosine. All right, so the way I always remember which are purines and which are pyrimidines is pyrimidines have a Y, and both our pyrimidines nucleotides also have a Y, so thymine and cytosine. Supposed to be E there. All right, our RNA both has, and our purines are going to be same, which is adenine and guanine. And then our pyrimidines, we're going to see one difference. So instead of thymine, you're going to have uracil, but you still will have cytosine. So we talked about in that chapter that one of the big differences between RNA and DNA is that DNA has thymine as a nucleotide, where RNA has uracil. So that's one of our biggest differences. And so anytime you're seeing genetic material, and if you see uracil in it, or if you see a U, because we're going to break these down and represent them by their first letter. So we're going to say A, T, G, and C, and then A, U, G, and C. So anytime you see any type of genetic material with a U, you know it must be RNA. And if it has a T, then you know it must be DNA. All right, so to draw a bit of what our DNAs here look like. So here we have our single nucleotides. And the only thing we're missing is our nitrogenous base is here. And so we're going to draw our adenine. So we're going to add a nitrogenous base that has a two-ring structure. I'm going to put an A there so we know that that's adenine. And then we're going to do our thymine. So thymine only has one ring structure. I'm going to do a T there. And then we're going to do a guanine. So guanine, once again, now has a two-ring structure. And then lastly, we'll do our cytosine, because we're making a DNA molecule, so we're not worrying about this as RNA right now. It appears my green marker is going bad. So there's our cytosine. All right, so here we have then, once again, our adenine, guanine, thymine, cytosine. Now, I drew these double ring structures, You might say, well, aren't they just the same? Shouldn't these both be adenine? But there are different other molecules like nitrogens and stuff like that that are on there that we're not drawing. And that's what actually makes these guys different and these guys different. So we're just drawing the basic ring structure just so we can get an idea that our purines and pyrimidines are different. Now, we need to talk a little bit about how we discovered the shape and the function of DNA. And this actually goes back to the early 1900s. So in the early 1900s, there was kind of this space race mentality. Now, if you ever think of the idea of a space race is after World War II, between the US and the USSR, the Soviet Union, there was this race to send someone up into space. And because of that, there's all this time and energy from scientists put in to make their country be able to first to send a rocket to space, and then man to space, and then all that kind of fun stuff. So there's a lot, so we call that the space race, right? Well, before there was a space race, there was the DNA race. And that's because out there, they were trying to figure out what is the genetic thing that controls all of the cell. And so they knew inside the cell then there was this nucleus, and inside the nucleus there was genetic material, or we didn't know it was genetic material at the time, but we had nucleic acids and we had proteins. Remember, we learned about how our nucleic acids or our DNA is wrapped around those histone proteins in our last chapter. Well, so we didn't know in the early 19th century whether or not it was the proteins that were doing the job of controlling the cell, or if it was the nucleic acids. So there were some basic experiments that ruled out proteins, and then at that point we knew that it must have been nucleic acids that were the ones that were controlling how the cell works. Because when we took the nucleic acids out of the cell, then the cells went into apoptosis, which is cellular death. But when we took the proteins out, it was still able to work and grow. So we knew that the genetic material must have been the nucleic acids. So then after that, it was this idea of how is DNA formed, and how does it actually work as this blueprint for everything in that cell. So they knew what the shapes of each of these nucleotides look like. And they actually even knew that they combined like this. So this is what we call our phosphate backbone. Well, we knew that they combined like this, but we didn't know how did it create that double helix, or even that the DNA was a double helix. And so back in the early 1940s, there was a young scientist named James Watson. He lived in LA, and he was really good with proteins. In fact, he would be able to look at something called an X-ray crystallography, and this is where they made X-rays of protein structures, and he'd be able to tell what that protein structure actually was based off these X-ray crystallographies. And he became very popular in this field. And then there was another scientist in London that was named Francis Crick, who was trying to figure out what the structure of DNA is. And so they both came together to be able to see if they could figure out how was the structure and form of DNA. And when he came together, one of the first things that he saw when he came over to London was that one of his, Francis Crick's colleagues, actually created a nice crystallography of DNA, and it looks something like this. And if you have your book, you should be able to see it in the book. It's obviously going to look better than what I'm drawing. So there was this crystallography of the DNA. And when Francis Crick showed this to James Watson, so it was Rosalind Franklin, who was the associate of Francis Crick that created this, and Francis Crick and Rosalind Franklin showed this to James Watson, and he immediately saw this and said, "Oh my gosh, this means from this picture I can deduce that DNA is double-stranded." So we've already talked about how DNA is double-stranded, but that all came from this crystallography. And not only that it was in a double helix. So we've talked a little bit about that double helix, and that's that twisted ladder
type of looking thing. And so they knew once they got together that DNA was going to be double-stranded. It was in this double helix. And so they started making all these wire models. And these wire models, they took all these nucleotides and they tried to put them together to mimic this double-strand, double helix DNA form. And the first thing they did was the other thing that they noticed, because this was symmetrical, almost looking like a sphere, they knew that this would be symmetrical here. All right, so you would have equal distance along this double helix. And one of the first things they did, they thought, okay, there's these phosphate backbones here. I wonder if those phosphate backbones could line up. Okay, so let's get rid of our crystallography here. So we're going to line up all these phosphate backbones, because that's going to make a nice kind of a helix. But what they realized is when you added another strand here, what it did is it made it so it looked more like this, and it wasn't uniform in distance. Some places it was closer together, others it was further together. And that's because you had these different nucleotides, whether they're purines or pyrimidines, having either two of the rings or one. And so it kind of made what we like to call the Coke bottle shape, right? So they immediately scrapped that idea. That didn't fit their model. So they flipped it around. They said, okay, well maybe then our nucleotides must interact with each other. But if our nucleotides with their nitrogen spaces are interacting with each other, then there's got to be some type of pattern. And so the first idea was, all right, well maybe the adenines match up with the adenines, the thymines match up with the thymines, the guanines, and so forth and so on. So when they did that, like, okay, perfect. Boom, boom, boom. Boom, boom, boom. But what they've noticed once again, by doing that, also created that Coke bottle shape, right? It wasn't linear. And so they immediately scrapped that idea. But then what that did tell them was, oh, this makes sense. If we have a purine on one side, then we need to have a pyrimidine on the other side. So that means there would be 3. Then there would be 3. And then 3. And 3. So by the time we put in our other things, what we see is a nice straight line, right? Because now in the middle of our 2 strands, we're going to have 3 rings on each one if you match up a pyrimidine and a purine. All right, they were getting somewhere at this point. But then they're like wondering, okay, does that mean that any purine can match up with any pyrimidine? So adenine can match up with thymine or cytosine. Guanine can match up with thymine or cytosine. So they decided to look more into it. And when they dug into the chemistry, what they found was something that was very interesting. And what they ended up finding was that here on our nitrogen spaces, there were certain places where they could do what we call hydrogen bonding. So we've talked a lot about hydrogen bonding with water. Water is not the only thing that can do hydrogen bonding. These guys could do hydrogen bonding, but only certain amounts per purine or pyrimidine. And so for like our adenine, it could do hydrogen bonding in 3 places. Same with our thymine. Our guanine could only do hydrogen bonding in 2 places. Well, guess what?
that was the same as the cytosine. And so they wondered, would it be possible that our second strand is actually attached together to the first strand from hydrogen bonding? And so they switched up their model, and what they noticed as well is that where the hydrogen bonding was, you couldn't have this facing up. Instead, it had to face the opposite direction. That marker's going bad. Let's try this one. Oh man, all my markers are going bad here. That comes the hard part, is drawing in. And so they made their model like this, showing that these ones are going in the opposite direction as these ones. And it fit perfectly with how the hydrogen bonding went between these different purines and pyrimidines. And so at this time, in 1953, they claimed to make the discovery that they knew how DNA was structured in this double helix form, and that our adenine will always match up with the thymine, and our guanine will always match up with the cytosine. And that's where we now say we do this, so we don't always draw DNA out like this. Instead, we'll kind of draw it out linear like this, and we'll say, okay, there's A, T, G, C, and then we know, okay, the double part of that, or the second strand of that, is going to be T, A, C, and G. And that is now our double helix, or our double-stranded DNA. And once they figured that out, they then realized, oh my gosh, this actually makes it really easy for us to understand how DNA would replicate. So we looked at in mitosis, meiosis, how there's a part where we had to replicate our DNA so that when we split our cells, we'd have DNA in both cells. Well, it'd be really easy, right? If we split this this DNA strand in half, we separated, we broke those hydrogen bonds, we could create another strand based off of that. Now we just created 2 new strands of that DNA molecule, right? That's called DNA replication. We're going to see here in a minute how this all worked. And so Francis Crick, James Watson, they published their idea and said, this is how we believe DNA is structured. And because of this, we now know that this is how DNA is also replicated in the cell. It's a really easy process. Now, this was all based off of theory using the chemistry and that crystallography graph. It wasn't until many years later that we had more advanced technologies that we could actually prove this theory that James Watson and Francis Crick came up with. And now we know that yes, they were absolutely right in that this is how DNA is structured, that they have this double helix, that we have a purine that matches up with the pyrimidine, and that we have specific ones. Adenines always match up with thymine, so A to T, and guanines always match up with cytosines, so G to C. Then we can also apply that to our RNA, but just throw out the thymine and put in the uracil. And what they did when they looked at the structure of uracil, yes, it was very similar to thymine, and it could do 3 hydrogen bondings, and so it also fit this narrative as well. And that is how James Watson, Francis Crick, and Rosalind Franklin were the 3 that discovered the structure and function of DNA at this very basic level. So what we're going to do next is now erase all this and show how this whole process of DNA replication actually happens with certain machines and stuff inside the cell. But we'll see that it does follow this nice pattern of being able to split the DNA in half and then add in the matching nucleotides, or what we call base pairs. So we call these base pairs. These are 2 bases, so they pair together, base pairs, and you just match those base pairs up. Now we're going to look at DNA replication, or sometimes we call it DNA synthesis. So if you remember back when we talked about the cell cycle, we had that S phase, and that S phase stands for synthesis. So that's why it's DNA replication or DNA synthesis. The whole idea is that we're making more DNA. And a good definition of that is the creation of 2 daughter strands from 1 parent strand. So we're going to have 1 original parent strand, and then at the end of it, we're going to create 2 new daughter strands. And so let's draw this out again. We're going to do this a little bit different. We're going to add multiple base pairs. So let's say our original strand looks like this. It's TACAGGAT. So we know the complementary strand is going to be A, T, G, T, C, C, T, A, right? Going back to our base pair theory here, that T's and A's always go together and C's and G's, so it's really easy. So what we're going to do is we're going to end up splitting our parent strand. So here's our parent. And we're going to split this in half. So here we have our hydrogen bonds holding this together. Remember we talked about hydrogen bonds, how they are very weak, but if you have a lot of them, they're strong. So since we have a lot of these hydrogen bonds holding this DNA together, it's strong, but it's also really easy to go in here and split that apart as well. All right, so we have T, A, C, A, G, G, A, T, and then A, T, G, T, G, G, T, A. And then what we can do, once again, we can add our base pairs by going off that knowledge. So I want you to say this with me. I know your parents are gonna, or your other family might think you're crazy, but say this with me. So T, then we're gonna have an A, T, G, T, C, C, T, A. And then here we're gonna have T, A, C, A. Oh, these are supposed to be C's. G, G, A, T. And so what we see with our DNA replication is that it is a semi-conservative replication, which means each new daughter strand, that should be strand, is made up of half the parent strand. So here's our parent strand. Boom, we've created our two new daughter strand, but we have conserved so partially conserved the parent strand in our two new daughter strands right here. All right. So that's what we mean by semi-conservative replication, is we're conserving some of the original DNA in half of our two new strands. All right, so there's actually, this is a little bit more technical than just doing it this way like I talked about. So we already kind of drew this out. So let's look exactly how that works. So when we draw our DNA like this, and we're not showing our A, T, C's, and G's, these lines represent our base pairs. So you can imagine there's an A and a T here, or a C and a G, right? We don't know exactly what it is, but we're just kind of showing it that way. So when we do it like this, we know it's in double helix form, and this is how we see our DNA majority of the time. But when we're going to replicate it, we're going to have to make it linear, and then we have a part in our DNA that is called the origin of replication. So within our DNA, there are specific codes that represent our origin of replication. And what that does is that allows a machine to come in here and start to split our hydrogen bonds holding our base pairs together to create kind of this bubble. All right, so then we would see here, then as our base pairs are separated out, T, A, C, A, G, G, all right, so we know that's T there. So we have an A here, we have a T here, we have a G here, A here, C here, C there. So we opened up our DNA, this origin of replication. So this little guy, what he's gonna do is he's gonna head this way to start to continue to open up our DNA so we can start to make our copy of it. And then we have this thing called our DNA polymerase. And the DNA's polymerase job is to actually add on those matching base pairs. So it's the machine that's grabbing so we have two G's here, so it's grabbing a C, and it's going to add that. It's grabbing another C, it's going to add that. And as it's getting ready now to go over the add mean, it knows it needs to grab a T, and it's going to start to add that. And our RNA polymerase is going to follow this guy right here as it continues to split and open up the DNA. All right, and as it gets to this part here, there's actually another actor right here. And its job is to unwind the double helix so that this guy can keep going through and cutting open the DNA, or cutting the hydrogen bonds and splitting open the DNA, so that this guy can continue to follow this RNA polymerase. So as it continues on, it will continue to add the base pairs. And it's just going to follow this guy, continually adding on the base pairs. So this top strand, we're adding on the complementary strand. Well, what about our bottom strand? Okay, good question. We also have a DNA polymerase that's doing the same thing here, and it's going to be going in the opposite direction. So it's going to add on a T and an A, and soon a C, and it's going to be heading the opposite direction. But there's one thing that's a little bit different, is that there's not a guy opening up this end. So when it gets here, it's actually going to be done, because what's going to happen is further down here, there's going to be another origin of replication where all of this will be starting and happening again. So eventually this is all going to open up, and once this all does open up, you'll have another one of these DNA polymerases start here and start to open up more this way, right? Because this guy's going down this way, and so you're going to have another DNA polymerase jump on here and then meet up and connect these two there. And this is what happens when we replicate our DNA. We talk about how much we have so much DNA in our cells. This is why you have multiple places of origin of replication. So you have replication happening on different ends of the whole DNA, so that this can all be done within a short period of time. When I say short period of time, it's still going to take, tens and stuff of minutes, so maybe up to like an hour or so. But if you were just to start at one end and try to replicate the whole thing, it would take hours upon hours and maybe even days to be able to replicate your whole DNA. But because you have multiple places of origin of replication, you can go in and start to replicate at different parts of your DNA, and as these parts meet up with each other, then you can combine all of the new DNA that you created, and at the end of the day, you're going to end up with two new daughter strands of DNA from our original one parent strand. So that's DNA replication. That's how it happens. Now, if we were in a more advanced biology class, we would talk about all these different actors and exactly how it works, but just know that we have, probably the main one you need to know is this DNA polymerase. All right? And its main job is to add that complementary strand to the original parent strand, whether it's this top one or this bottom one. And like I said, they're going in opposite directions. All right. So these other guys, topoimerase, splicease, all these other things that are in here, we're not going to get too deep into them, but know that there's multiple machines allowing this to happen to replicate our DNA. So that's DNA replication. And the one thing that's really important to realize, DNA replication happens in the nucleus. And the reason why that's important to know is that we keep our DNA in our nucleus. It's our most prized possession for our cells. And so we don't want it to leave the cell. So we keep it in the nucleus. And so DNA replication only happens inside our nucleus of the cell. All right, so the next part we're going to be talking about is RNA transcription. So when we looked at our cell, we talked about how there was a signal given to the nucleus, and the nucleus would then copy a gene into an RNA form. That RNA form would go out to the ribosome, get translated, and then we make a protein. So we're now going to see how that process works. How do we take a gene that's in the DNA, create a RNA copy of that, so the RNA copy can leave the nucleus, go find a ribosome, be read and translated by the ribosome, and create the protein that we need from it. So that's going to be the next part of this chapter. Okay, so the next part of this is understanding how do we go from our DNA to our protein. And this is something we call the central dogma of molecular genetics. So most of you may or may not know what dogma means, but dogma is kind of like, so if you think of like religion, it's like, what's the most important part of a religion, right? If you were to explain like Christianity, and if you ask somebody, tell me what's the most important part of Christianity, you might say that the most important thing is that Jesus died for our sins on the cross, right? That would be kind of like your most basic breakdown of what Christianity is. Now, there's a whole bunch more to Christianity, but that's kind of its basic breakdown. And that's the same thing here when we look at the central dogma of molecular genetics. Its central idea is that you go from a DNA code to an RNA transcript to a protein. And that's what we're going to be seeing for the rest of this chapter, is how do we go through this process of going from a DNA to an RNA to a protein. That's the central dogma of molecular genetics. So the first thing we're going to look at is RNA transcription. So that's that first step, going from a DNA to an RNA. So copying a DNA gene into an RNA transcript. All right, so to do this, we're going to draw out our DNA. Realize that this is going on for, not forever, but for a long time. This is going on for a long time. So we're just looking at this specific spot of our DNA, and specifically what we're going to call our gene of interest. So this area of our DNA codes our gene of interest. So the gene we're trying to make a copy of because it codes for a protein that our cell wants to create. Now there's going to be different parts to this gene of interest, and this first part here is going to be called our promoter. And then another part is going to be our termination sequence. Let's not use that. Let's go with maybe that. So our promoter tells our machines, hey, this is the very first part of our gene. This is where our gene starts. And then our termination sequence tells our machines, hey, this is the end of the gene. This is where the gene ends. And so we know this from our promoter to our termination sequence is where we find our gene of interest. And so what happens is that promoter shows the spot right where our next polymerase, so we just saw the DNA polymerase, now we're going to have what we call an RNA polymerase. And these are much bigger than DNA polymerase, they take up the whole double strand. Okay, so we have our RNA polymerase, and then it's going to go down here, and as it goes down, it's going to create a copy of our gene on one strand. So remember, if we split this up, we have two strands, but the gene is only, the code is only found on that, for this part, going to be our top strand. All right, so we're going to open that up. Like I said, as it does that, actually let's take kind of like a glimpse here. So when that polymerase is over that specific area of our DNA, it has opened up our DNA. So once again, let's say this is T, A, C, A, G, G. Go T again there. That means this would be A, T, G, T, C, C, A. But our code is coded in this top strand here, and so our polymerase is only going to add the matching base pairs to that top strand. And now, since we're creating an RNA instead of a DNA, we have to make sure we're putting the right base pairs on there. So T will always still match up with A, but A now is going to match up with U. Remember, you're a cell. This is how we know this is RNA. Then we're going to still have G, then we're going to have U again, then we're going to have C, C, and A. And once again, we're only making a copy of this top strand. So we're not worried about this bottom strand because the code is found in this top strand. I'm not going to write out these letters, but then you can see as it's moving here, the DNA strand is going to end up coming out the end. And our DNA, once it comes out, our DNA is going to come back together. So our RNA strand, sorry, I said that wrong. Our RNA strand is going to come out, and then our DNA is going to come back together as this goes down. So pretty much you can kind of see it like this. And as it moves along here, it's going to have a longer and longer RNA. And then when it gets to our termination sequence, this termination sequence tells our RNA polymerase, hey, we're done, this is the end of the gene, let's break off.
And so that's what ends up happening. Our polymerase breaks off here, and then our RNA transcript also breaks off. And I'm just drawing these half lines. We could put in AT, not AT, we could put in AUG, whatever it is, but we're not going to do that for now, just to make this simplified. And so we see here, and then as it moves on, like I said, that DNA is put back together, and by the time it's all done, you still have just this nice DNA strand. And if we need to make another copy, that RNA polymerase can jump back onto this promoterase and make another copy of that RNA transcript if needs be. But that is our RNA transcription. That's how we make a copy of that gene that is stored inside the DNA, and we make that RNA transcript of it. Now, what happens to this RNA transcript after it's copied? You're probably thinking, okay, it's going to leave now the nucleus and go find a ribosome. Yes, it's going to do that, but it actually has to go through a couple more steps, 3 steps to be exact, before it's ready to leave the nucleus. And that's what we're going to see next. All right, so we just went through RNA transcription. So we took our DNA gene and we transcribed it into our RNA. But before it can leave our nucleus, so here's our nucleus, before it can leave our nucleus and go out to find our ribosome to be translated, it needs to go through something we call RNA processing. So there's 3 specific things that need to happen to our RNA. And the first one is we actually have to trim our RNA up. We have to take out our introns and leave our exons. Okay, so our whole DNA transcript here doesn't have everything it has more than what we need for our gene to create our protein. And some of these spots are called introns. All right, so these here are introns. And so what we have is we have these little machines, once again, that are going to go in and cut out those introns. So we're going to take out those introns. And everything that's left after that, so our RNA transcript's now going to be a little bit smaller. Everything that's left after that now is going to be what we call our exons. And so one way we always remember which one are introns and exons. So our exons are going to go and be expressed into our protein. So exons expressed, therefore the introns are the ones that are taken out. So that's our first step. Our second step is that we have to add on our poly A tail. So what we do is we add on a whole bunch of adenines to the very end of our transcript, our RNA transcript. And so we write these as AAAAA, and there's a whole bunch of these. And the reason why we do this is this kind of acts as a time clock on how long this transcript is going to last out into the cell.
So we've always talked about how we make sure that we keep our DNA inside our nucleus. And one of the reasons for that is we have enzymes that patrol the rest of the cell, and anytime it finds genetic material, it starts to break down that genetic material from the outside in. And so therefore, if our DNA gets out into the inside of the cell, it's going to start to be broken down, and then we have nothing to control the cell, and the cell's going to go into death, apoptosis. All right? And so that's why we always keep our DNA in the nucleus. And the reason why we have these enzymes out here that break down DNA particles is to help protect our cells from foreign invaders, mostly viruses and also bacteria as well. So if a virus inputs its DNA into a cell, there's a good chance that those enzymes are going to go and break down that genetic material of that virus, and now our cell is saved. All right, so because of that, we have to add in these poly-A tails. So these poly-A tails they're not part of our gene code. Instead, once our transcripts gets out into the cell here, and if an enzyme starts to eat at it, it's going to slowly break down this transcript. So it kind of puts a time clock on this, because by the time it finally gets to here, then it starts to get into the gene, and now that RNA sequence is done and it's no longer needed. So in other words, if you're trying to make a lot of protein, you're going to want to have this transcript translated multiple times, so you're going to put on a lot more poly-A tail so it lasts longer inside the cell. If you want it to only do a little bit, you're going to do a smaller poly-A tail. So once again, this protects it, but also kind of gives this time clock on how many times our RNA transcript is actually translated. All right, so that's our second step. Our third step in our RNA processing is we have to add a 5' cap. So on the other end here, this is what we call our 5' end, and that's all based off of whether it's going up or down on our nucleic acids when we showed them at the double helix. So this is our 5' end. So this little dash represents a prime. This over here is actually our 3' end. So those are the two ends of any type of genetic material. There's a 5' end and a 3' end. That's how you know if it's going up or down. So this is our 5' end, so we add a 5' cap. Now what this 5' cap does, this also protects our genetic material from getting eaten up by those enzymes. So in other words, the enzymes can't eat up from this end because this 5' cap is protecting it. It can only eat up from that end. This is why your time clock is your poly-A tail. And then the other thing that this does, two other things. One is it kind of acts like the security card to get in and out of the cell, the nucleus, mainly getting out of the nucleus because it doesn't need to come back in. So this is a security card for it to allow it to leave the nucleus. And then at the very end, we're going to see when we look at translation, this plays an important part in translation as well. So these are the three things that happen. The first one is we take out the introns and add and leave the exons. Second, we add poly-A tail. And third, we add our 5', so 5' cap. All right, so these three things, we refer to them as RNA processing. So the processing of the RNA before it can finally leave. Once it's now ready to leave, we actually call it by a different name. We don't call it an RNA transcript anymore. We now call it a mRNA. And we do this a lot with RNAs where we give them another small letter number in front of it to represent that it's a certain type of RNA. So our mRNA stands for messenger. So once we go through all this processing of the RNA transcript, we now call it an mRNA, and now it can leave out of the cell to go find the nucleus to go find a ribosome to